This video will go through a step-by-step animation of the LogD method currently in place at the Drug Discovery Unit, University of Dundee. The pharmacokinetic properties of a drug, its solubility, absorption, distribution, metabolism, binding and final excretion from the body are governed in part by the intrinsic lipophilicity of the molecule. Lipophilicity itself is defined as the affinity of a substance for a lipid phase, or the organic phase. It can be defined by the partition coefficient P. The log 10 of P is usually used due to the large P values. The equations for both are detailed below. The higher the affinity for the organic phase, the more drug will partition to it, and the higher P or log P values. For ionizable compounds, both the ionized and unionized forms will partition between the immiscible liquids, and the distribution coefficient, also known as D, can be defined. The log to the base 10 is also usually used and gives us log D. The equations for the distribution coefficient and log D are detailed here. The log D of a compound can be predicted using a fast gradient reverse phase HPLC method to determine the chromatographic hydrophobicity index, also known as Chi. Here the term hydrophobicity is used as the HPLC method is not a simple binary solvent partition, but has a direct relationship to lipophilicity. The Chi of each compound, which is a measure of the percent of organic phase concentration by which a compound is eluting from a column, can be measured at 3 different pH values, namely 2, 7.4 and pH 10, which can help aid in the determination of the acid-base nature of compounds as well as neutrals. For this experiment, you will need ammonium acetate, deionized or milk water, acetonitrile, 10 millimolar test compounds in DMSO, 1 mg per ml calibration mix. The table to your right details each of the calibration standards, which should all be in the mix. V bottom 96 well microtiter plates, Eppendorf tubes, HPLC vials, 15 ml centrifuge tube or vials, pipettes capable of dispensing 10 and 200 microliters and 1 ml, plus suitable tips. High or ultra performance liquid chromatography system, HPLC or UPLC. First, we're going to prepare our solutions. The first solution we're going to prepare is our 50-50 acetonitrile water. Prepare this solution and store at room temperature for up to 3 months. Now we can prepare our calibration mix. First, create 1 mg per ml stocks of each of the calibration standards. Aliquot 100 microliters of each calibration standard into the same centrifuge tube or vial. A table of the calibration standards is listed here. Add 9 ml of 50-50 acetonitrile water to get 10 micrograms per ml final concentration. Finally, sample into 400 microliter aliquots and store at -20 degrees Celsius for later use. The calibration standards can be stored and used for up to 6 months. Preparation of uracil, the T0 marker. Create 1 mg per ml stock of uracil in DMSO. Dilute 10 micrograms per ml in 50-50 acetonitrile water. Finally, sample into 400 microliter aliquots and store at -20 degrees Celsius for later use. These aliquots can be stored and used for up to 6 months. Uracil has a very low retention time, close to 0, in reverse phase chromatography, and is used as our T0 marker in the assay. This is used later when calculating retention factor for the standards and samples. Now we can prepare our ammonium acetate buffer. Prepare a 1 molar solution of ammonium acetate. This can be stored at room temperature for up to 1 month. Next, prepare 50 millimolar dilution from the 1 molar stock. Adjust to pH 7.4 with formic acid or ammonium hydroxide solution as necessary. This can also be stored for up to 1 month. This buffer can be made to a different pH depending on the experimental needs. For this example, our experiment is carried out at pH 7.4. Preparation of compounds. Prepare each compound to 10 millimolar stock in DMSO. Dilute compounds to 0.25 millimolar by taking 5 microliters of the 10 millimolar stock into 195 microliters of 50/50 acetonitrile water. Now we can set up our analysis. Set up the UHPLC or HPLC method. This assay can be run using either instrument. Example conditions used in the Drug Discovery Unit Dundee are above. Here we use a UHPLC system with a suitable column. Once the HPLC method is set up, you can now inject your calibration standards and a uracil T0 marker and samples onto the HPLC. Blank controls containing 50/50 acetonitrile water, 50/50 acetonitrile buffer, and the calibration standard mix should be analysed at the start and end of each run. If the number of test samples exceeds 30, further controls including blanks and calibration standard mix should be included. Data analysis. Calibration standards. Our calibration standards have a range of known chi-naught values published in the paper below. These are detailed in our table here. For example, our standard theophylline has a chi-naught value at pH 7.4 of 18.4. We will use these chi-naught values in our further calculations. First, let's calculate the retention factor, or K, of each standard using the following equation, where retention time of standard minus retention time of uracil divided by the retention time of uracil. Here you can see that our uracil retention time is 0.3 minutes. So now let's apply this to our first calibration standard, theophylline. So the retention factor, or K, for theophylline is the retention time of the standard, which is 0.91 minutes, minus the retention time of uracil, which is 0.3 minutes, divided by the retention time of uracil. This gives us a retention factor of 2.03. Now do this for the rest of your calibration standards. Once retention factor has been calculated for each of the standards, we can then create a linear regression curve of retention factor versus chi-naught at pH 7.4. Here is our calibration line, displaying the equation of the line and the R-squared. Typically, the R-squared value for our equation of the line should be greater than 0.9. In addition, up to 3 points can be taken out of the calibration line if necessary. This is at the analyst's discretion. We can now calculate chi for each standard using the above formula and the equation of the line. For a linear regression curve, the equation of the line follows the formula of y equals mx plus c. For our line, our equation is y equals 0.0811 x plus 0.5466, where our slope is 0.0811, which is also called m, and our intercept, or c, is 0.5466. To calculate chi, or x, we need to rearrange our equation of the line to follow the formula of x equals y minus c divided by m. So to calculate chi equals retention factor minus intercept divided by slope. Let's put this equation into practice for our first calibration standard, theophylline. For theophylline, chi equals retention factor, which is 2.03, minus the intercept, which is 0.5466, divided by the slope, which is 0.0811. This gives theophylline a chi of 18.3. Now do this for the rest of the calibration standards. Finally, we can calculate our chi log D at pH 7.4 for each standard. To do this, we use the following equation: chi log D equals 0.054 times chi minus 1.467. This comes from a paper from Valco, which discusses the lipophilicity and biomimetic properties measured by HPLC to support drug discovery. This equation is based on the fitted data of 98 known drug molecules measured for chi and log P values. Many structure lipophilicity relationships have been computed for in silico prediction of partition coefficient, also known as log P. Hence, medicinal chemists prefer lipophilicity scale that is comparable to the log P scale. Therefore, the chi-scale of lipophilicity has been introduced to convert to a chi log D measurement, which we are going to use to calculate chi log D at pH 7.4 for each standard. Now let's put our chi log D equation into practice with our first calibration standard, theophylline. To calculate chi log D equals 0.054 times our chi value previously calculated, which is 18.3, minus 1.467. This gives theophylline a chi log D value of negative 0.5. Now do the same for the rest of your calibration standards. Once the calibration standards have been calculated, we can now follow the same steps and calculate the chi log D results for our unknown samples. Step 1 is to calculate the retention factor for each unknown compound, where retention factor equals the retention time of the unknown compound minus the retention time of uracil or T0 marker divided by the retention time of uracil. For our example, the retention time of uracil is 0.3 minutes, and the retention time of our unknown compound 1 is 1.5 minutes. So for example, retention factor for unknown compound 1 equals 1.5 minutes minus 0.3 minutes divided by 0.3 minutes. This gives a retention factor of 4. Do this for the rest of your unknown compounds. Step 2 is to refer to the calibration line previously created from the calibration standards. Step 3. Use the equation of the line to calculate chi, where chi equals retention factor minus intercept divided by slope. From our line, our slope is 0.0811 and our intercept is 0.5466. Now for unknown compound 1, chi equals retention factor, which was previously calculated as 4, minus our intercept at 0.5466 divided by the slope, which is 0.0811. This gives a chi value of 42.6. Now do the same for all other compounds. Finally, calculate chi log D for each unknown compound. Chi log D equals 0.054 times chi minus 1.467. So for unknown compound 1, chi log D equals 0.054 times 42.6 previously calculated minus 1.467, which gives a chi log D value of 0.833 for unknown compound 1. So we have now calculated our chi log D values for all of our unknown compounds. So what do these chi log D values actually mean? Here we have a table of LogD at pH 7.4 and pharmacokinetic effect. You can see on the table that LogD value of 1 to 3 is an indication of ideal drug candidates. With possible good oral absorption, solubility tends to remain good, low metabolic clearance and good CNS absorption. However, it is not always one size fits all, and this table is just a guideline.